Okay, so let's dive in, right? We're tackling something pretty huge today, a real shift in how we understand biology, actually.
Absolutely. It really goes back to the human genome and what we thought we knew versus what we actually found.
Right. For ages, it was all about the proteins, wasn't it?
Right.
That was the core instruction manual, the protein coding genes.
Exactly. And, well, biochemically, that made a lot of sense. Proteins do everything, right? They're the enzymes, the building blocks.
The messengers.
All of it. But then the Human Genome Project finished, and the data just presented this massive mystery. It really changed everything.
And that mystery is just sheer numbers, isn't it? We map 3.2 billion base pairs. This incredible amount of information.
Yeah, a phenomenal achievement.
But then you find out only, what, 1.5% of that actually codes for those proteins we were so focused on?
Just 1.5%. It's tiny.
Yeah.
So the obvious question was, well, what's the other 98.5% doing? Is it just there?
And that's where that term genome dark matter came from.
Okay.
Or
Even junk DNA. Yeah, that was a really common term for a while. The idea was maybe it's just evolutionary clutter, leftovers.
But as our sources make really clear, that's absolutely not the story. It's not junk. It's not filler.
Not at all.
Yeah.
We now know that huge chunk, that 98.5%, it's almost entirely dedicated to regulation, to control. Some analyses suggest like 80% of the whole genome is involved in regulation somehow.
80%. That completely flips the script. It's not about having more parts.
Yep.
It's about having a much, much more sophisticated plan for using those parts.
Precisely. Think about complexity. Why are humans so much more complex than, say, a simple worm?
Right. The source material uses that comparison.
Same.
C. elegans, the nematode worm.
Yeah. We have roughly 20,000 protein-coding genes. That worm has about the same number. Often they're even the same basic genes, the homologs.
So same materials, essentially.
Pretty much the same construction material. So why are we? Well, us, and the worm is a worm.
It has to be the planning, the architectural blueprints.
Exactly. That's what the non-coding regions provide. The when, the where, the how much, for every single one of those 20,000 genes. It controls the expression, ramps it up, shuts it down.
The whole orchestra, basically. The conductor.
Perfect analogy. The difference is the orchestration, not just the players.
And that brings us right to the stars of our show today, the non-coding regulatory RNAs. These are the molecules made from that dark matter.
They get transcribed from DNA, just like protein-coding genes.
But crucially,
They are never translated into protein. Their job is finished as RNA. They are the regulators themselves. And it's not a small fraction. Over 60% of our genome gets transcribed into these non-translated RNAs.
60%. Wow. Okay, so they're clearly central.
Absolutely central. And this is why, anyone interested in health and disease really needs to pay attention.
Because the variations linked to disease aren't always where we thought they'd be.
Exactly. When scientists started mapping genetic variations, polymorphisms, linked to complex diseases, think diabetes, heart disease, and especially cancer, they found something surprising.
Where were the variations?
Overwhelmingly, they weren't in the 1.5% protein-coding regions. They're in the 98.5%, the regulatory regions.
So the problem isn't necessarily a broken protein part.
Often, no. It's more likely a problem with the instructions. The timing's off, the volume's wrong, the regulation of the gene is messed up.
And that faulty regulation might be a bigger driver of disease than, a structural flaw in the protein itself.
It seems increasingly likely, yes. And that's why there's this huge research push. Which brings us to our mission today, really.
To explore these regulators.
Yeah, specifically the two best understood types. First, the small but really powerful ones, micro-RNAs or miRNAs, the silencers.
And then the big words.
And then the big ones, the scaffolds and guides, long-coding RNAs or lncRNAs.
Okay, let's start small then. miRNAs. You said they're tiny, but this is where it gets really fascinating, right? The power they wield.
Immense power. Micro-RNAs are fundamentally fine-tuning silencers. And yeah, they're defined by being small, on average, only about 22 nucleotides long.
Just 22 letters of the RNA code. That's incredibly short.
It is. But don't let the size fool you. The genome isn't just dabbling here.
Right. I saw the number. The human genome has codes for almost 6,000 different miRNA genes.
Almost 6,000. That's nearly a third of the number of protein-coding genes. So it's this huge parallel regulatory system operating alongside the protein world.
An entire network just for control.
Exactly. And what's really striking is how old and fundamental this system is. It's not some recent evolutionary invention.
It's conserved, right? Found everywhere.
Deeply conserved. This mechanism, post-transcriptional silencing by miRNA, you find it in basically all eukaryotes, plants, animals, fungi. It's fundamental to complex life.
And even bacteria, I thought they were simpler.
Well, they are simple overall, but fascinatingly, even bacteria have a sort of primitive version of this machinery. They seem to use it mainly as a defense system.
Defense? Against what?
Against foreign DNA, like from viruses, phages, that try to inject their genetic material. The bacteria can recognize and silence that foreign stuff using RNA interference.
Wow. So it's an ancient defense mechanism that got co-opted for internal regulation in more complex life.
That seems to be the story, yeah. A
Yeah.
core biological strategy.
Yeah.
So to really get how they work, we need to walk through the mechanism, this post-transcriptional silencing pathway. It's like a little molecular assembly line.
Okay, take us through the line. Where does an RNA start its journey?
It starts in the nucleus, like most gene products, and miRNA gene, sitting in that non-coding DNA, gets transcribed. This produces a longer primary transcript called miRNA.
Okay, so a raw transcript first. It needs processing.
It does. It gets processed in a couple of steps, partly in the nucleus, then it's exported out into the main cell body, the cytoplasm, as a precursor molecule.
And that's where the key tool comes in. You mentioned dicer.
Exactly. Once in the cytoplasm, this crucial enzyme, dicer, takes over. Dicer is a type of nucleus, meaning it cuts RNA. It recognizes a specific shape in the precursor miRNA and trims it down.
So dicer is what creates that characteristic short length, around 22 nucleotides?
Precisely. Dicer makes that final precise cut, producing a short, double-stranded RNA fragment.
Double-stranded. I thought RNA was usually single-stranded.
It often is, but this intermediate is double-stranded. But then one of those strands gets loaded into another key player,
Which is?
a big protein complex called RISC. That stands for RNA-induced silencing complex.
RISC. Okay, so dicer cuts, and then one strand goes into RISC.
That's the crucial step. That single strand, the mature miRNA, becomes the guide within the RISC complex. Think of RISC as the enforcement machinery, and the miRNA as the targeting system. It tells RISC where to go.
So RISC on its own doesn't do much. It needs that miRNA guide.
Exactly. The ncRNA uses its sequence, its 22 nucleotide code, to scan the cell's messenger RNAs, mRNAs. Those are the transcripts carrying protein instructions.
Looking for a match.
Looking for a complementary sequence. Based on base pairing rules, A with U, C with G, the miRNA guides RISC to its target mRNA.
And then what happens when RIC finds its target?
Well, there are two main outcomes, and it usually depends on how good the match is between the miRNA and the mRNA target.
Okay, outcome one. Let's say it's a perfect or near perfect match.
If the base pairing is highly complementary, almost perfect, RISC takes decisive action. It acts like molecular scissors and cleaves the target mRNA. It cuts it, usually right in the middle of the matched region.
And cutting it means?
It means the mRNA gets rapidly degraded, destroyed. The cell can't use it to make protein anymore. The message is just erased. This is actually very common in plants.
Total destruction. Okay, what about outcome two? What if the match isn't quite perfect, which you said is more common in mammals, like us?
Right. In mammals, often a miRNA only matches a specific short seed region on the mRNA target, maybe 6 to 8 bases, plus some surrounding weaker matches. It's an imperfect pairing.
And in that case?
In that case, RISC usually doesn't cleave the mRNA. Instead, it leads to translational repression.
Meaning
RISC essentially sits on the mRNA and physically blocks the ribosomes, the cell's protein-making factories, from translating that mRNA into protein. Or it might shuttle the mRNA off to be degraded more slowly.
So the mRNA molecule might still be physically there?
For a while, yeah.
But it's effectively silenced, muted. No protein gets made.
Exactly. The end result is the same in both cases.
Yeah.
The amount of the target protein produced goes down, either by destroying the blueprint or by stopping the factory from reading it.
Okay, that makes sense. But what gives these tiny miRNAs such leverage? You mentioned they have disproportionate power.
That's the crucial point. It's about scale.
Yeah.
A single type of miRNA doesn't usually target just one mRNA.
You can hit multiple targets.
Oh, yeah. A single miRNA can have binding sites, perhaps imperfect ones, on hundreds of different mRNAs.
Hundreds, from one little 22 nucleotide RNA.
Correct. As long as those mRNAs contain the right complementary sequence, that one miRNA loaded into RISC can dial down the expression of that whole set of genes simultaneously.
So it's not just fine-tuning one gene, it's coordinating an entire program of genes, like a master switch.
Precisely. It can co-regulate entire pathways, entire cellular responses, just by tweaking the levels of dozens or hundreds of proteins all at once. That's where the real power lies.
That's slightly terrifying, though. If one little molecule has that much control, what happens if it goes wrong? If it's overactive or targets the wrong things?
Well, that's exactly what we see in many diseases, especially cancer. Dysregulation of a single miRNA can flip a major cellular switch, like turning off growth control, leading to proliferation.
Because it's silencing a bunch of tumor suppressor genes, maybe?
Or maybe it's failing to silence oncogenes it normally keeps in check. That high leverage makes them incredibly important in pathology, but it also, flip side of the coin, makes them really attractive therapeutic targets.
Right. And this whole natural silencing mechanism, it didn't just teach us about biology, it gave us a tool, right? You mentioned CRNAs.
Exactly. Small interfering RNAs, or CRNAs, these are basically synthetic, lab-made mimics of miRNAs.
Designed by researchers.
Yes, scientists designed short, usually double-stranded RNA molecules specifically to target a gene they want to study. You introduce these CRNAs into cells.
And they use the cell's own machinery.
They hijack the exact same pathway. Dicer processes them, they get loaded into RASC, and then RASC uses the synthetic CRNA sequence as a guide.
To find and silence the target mRNA the researcher is interested in.
Correct. And because CRNAs are often designed for a perfect match, they usually trigger that very efficient mRNA cleavage and destruction.
And that's the basis of knockdown technology.
That's it. It's fundamental in labs everywhere. If you want to know what gene X does, you design an CRNA against its mRNA, knock down its expression, and see what happens to the cell. It's a way to functionally delete a gene's activity temporarily.
Which, of course, immediately suggests using this therapeutically. If you can knock down a gene in a lab dish.
Can you knock down a disease-causing gene in a patient? That's the therapeutic promise. If you have an overactive oncogene driving cancer, could you design a CRNA therapeutic to specifically target and destroy its mRNA?
Silencing the bad actor directly.
Selectively turning it off, without needing to permanently change the DNA itself. It's a very powerful concept.
Okay, that covers the miRNAs, the short, sharp silencers acting mainly in the cytoplasm. But now we shift gears, right, to the other major players, long non-coding RNAs, lncRNAs.
You do, and we shift scale dramatically. If miRNAs are the fine-tuners, lncRNAs are more like the architects and the structural engineers of the genome, and they operate primarily back in the nucleus.
Defined mainly by size, over 200 nucleotides.
That's the basic definition, yes. Anything non-coding over 200 nucleotides gets lumped into this category. But it's a hugely diverse group in terms of structure and function, much more so than miRNAs.
And the sheer number of them is staggering, isn't it? Even more than miRNAs.
Oh, vastly more. The estimates are still evolving as we sequence deeper, but some calculations suggest that the number of distinct lncRNA transcripts in our genome might outnumber protein-coding mRNAs by like 10 to 1, maybe even 20 to 1.
20 times more lncRNAs than protein blueprints. That's mind-boggling. The regulatory complexity is immense.
It's a vast, largely unexplored universe of regulation. We're really just scratching the surface of what most of these lncRNAs are doing.
And their main job, unlike miRNAs targeting mRNA, is focused on the DNA itself, on the chromatin.
Primarily, yes. Their main game is modulating gene expression by influencing the physical structure and accessibility of chromatin, that tightly packaged complex of DNA wrapped around histone proteins.
Okay, so remind us quickly about chromatin, tight versus loose.
Right. Think of DNA like a very long thread. To fit inside the nucleus, it's wound around protein spools called histones. When it's wound really tightly, that's called heterochromatin. The genes in that region are generally switched off, inactive, because the machinery can't get access.
Packed away.
Packed away. When the chromatin is looser, more open, that's euchromatin, the genes are accessible and can be transcribed. They're active.
And lncRNAs are controlling how tightly that thread is wound.
They play a major role in directing that process. They guide the enzymes that chemically modify the histones or the DNA itself, dictating whether a region should be open or closed for business. They're controlling the physical packaging.
Wow. Okay, so how do they do that? The sources mentioned 4 main ways they act, like 4 job descriptions for lncRNAs.
That's a good way to think about it. 4 primary mechanisms of action.
Let's take the first one, gen activation. How does an lncRNA promote transcription, acting as a facilitator?
Right. In this role, an lncRNA might bind near a gene's promoter, the on switch, and help stabilize the binding of transcription factors.
Those are the proteins that actually kick off transcription.
Exactly. The lncRNA can act like a scaffold or a platform, helping to recruit and hold those activating factors in place, making transcription more efficient or more likely to happen. It greases the wheels for activation.
Okay. Mechanism 2 is the opposite. Gene suppression, acting as a decoy.
Precisely. An lncRNA might contain a sequence that mimics the binding site for a specific transcription factor, or maybe even another regulatory RNA, like an RNA.
So it intercepts them.
It acts like a sponge or a decoy. It binds up those activating molecules, sequestering them, preventing them from reaching their actual target gene. So the target gene never gets the on signal.
Clever. It's not silencing the gene directly, but silencing the activator.
Exactly. It mops up the activators.
Okay, mechanism 3 sounds really complex. Promoting chromatin modification? This is where they're directing the winding and unwinding.
This is arguably their most profound role. Here, the lncRNA acts as a guide or a tether. It binds specifically to a certain region of the chromatin.
How does it know where to bind?
Through sequence complementarity with the DNA, or by recognizing specific structures. Once it's bound to a location, it then recruits the chromatin-modifying enzymes, the writers and erasers of epigenetic marks.
Like the enzymes that add or remove those chemical tags on histones. Acetyl groups, methyl groups.
Exactly. Acetylases, methylases, demethylases. The lncRNA acts like a beacon, bringing the right enzyme to the right address on the genome.
So if it brings in acetylases, that usually loosens the chromatin.
Generally, yes. Acetylation tends to open up chromatin, promoting gene activation, euchromatin.
And if it brings certain methylases?
That often leads to tighter packing, gene silencing, heterochromatin. The lncRNA is providing the targeting specificity for these powerful epigenetic modifications. It's directing the whole process.
That's incredible coordination. It's translating the linear code of the RNA into a 3D structural change in the DNA packaging.
Precisely. And the fourth mechanism is related, acting as a scaffold.
A physical structure.
Yes. lncRNAs aren't just floppy strings. They can fold into complex 3D shapes. These shapes can then serve as platforms or scaffolds to bring together multiple proteins into a functional complex.
Holding the machinery together.
Holding it together in the right way, in the right place. These assembled complexes might then modify chromatin or regulate transcription directly. The lncRNA provides the structural framework for the regulatory machine.
Okay, those four mechanisms, facilitator, decoy, director, scaffold, give us a sense of their versatility. And there's a classic example, right? XST.
XST is the textbook case. It's a fascinating and essential lncRNA. It's transcribed from the X chromosome.
And it's crucial for X inactivation in females.
Absolutely essential. Female mammals have two X chromosomes, XX, while males have one X and one Y. Having two fully active X chromosomes is actually toxic to the cell.
Too much gene product.
Right. So very early in female development, one of the two X chromosomes in every cell gets almost completely shut down, inactivated.
And XST is the molecule that does the shutting down.
It orchestrates it. The XXT lncRNA is transcribed only from the X chromosome that is destined to be inactivated. And then, remarkably, it doesn't diffuse away. It spreads out and physically coats that entire X chromosome like a blanket.
It paints the whole chromosome.
Effectively, yes. This XST cloak then recruits all the silencing machinery, the chromatin modifiers we talked about, leading to chromosome-wide gene silencing. It's a massive silencing operation directed by one lncRNA.
That perfectly illustrates that large-scale architectural control. Just incredible.
It really is. And it highlights how lncRNAs can act over huge genomic distances. One other point connecting lncRNAs to structure is their link to enhancers.
Enhancers those DNA sequences that boost gene activity, sometimes from far away.
Exactly. It turns out that many of these enhancer regions are actually sites where lncRNAs are actively being transcribed.
So the enhancer region itself makes an lncRNA. That seems purposeful.
It strongly suggests that part of how enhancers work might be through the lncRNAs they produce. These enhancer-derived lncRNAs might be involved in looping the DNA to bring the distant enhancer close to the gene promoter, or in recruiting activating factors. It reinforces their role as long-range coordinators.
Okay, so we've got the miRNAs as the fine-tuning cytoplasmic silencers and the lncRNAs as the nuclear architects and scaffolders. We started out saying that 98.5% of the genome wasn't junk, it was regulatory gold. So bringing this all together, what does this deep dive into the architects mean for, well, for medicine, for patients? Where's this knowledge taking us clinically?
Well, the potential is enormous, but so are the challenges. Because these RNAs are so fundamental to how cells operate, they're inevitably involved when things go wrong.
In development, in disease.
Absolutely. Malignancy, developmental disorders, neurological problems, cardiovascular disease. Dysregulation of miRNAs and lncRNAs is implicated in almost everything, which naturally makes them very hot targets for drug development.
But it's a different kind of drug target, isn't it? We're not aiming for one faulty protein anymore.
No, the idea is to target the faulty regulation itself, to fix the master instructions, not just cope with the downstream consequences. And one of the first big payoffs has been in diagnostics.
Using them as biomarkers.
Exactly. A really useful feature of these RNAs is that they're surprisingly stable, and they get released from cells.
Into the bloodstream.
Into the bloodstream, urine, saliva. They often get packaged into tiny protective vesicles called exosomes, so you can detect these circulating miRNAs and lncRNAs non-invasively.
And their levels or types change depending on what's happening in the cells they came from?
Precisely. A stressed cell, a cancerous cell, an inflamed cell, it releases a different signature of RNAs into the circulation compared to a healthy cell.
Which could give us an incredibly early warning sign for disease, long before symptoms appear.
That's the hope, and it's starting to happen. Our sources confirm that specific miRNA profiles in blood are already being used, for instance, to help classify different cancer subtypes which can guide treatment, or to detect early signs of heart damage. It's like a liquid biopsy, giving you a window into cell health without needing tissue.
Okay, that's diagnostics. What about actually treating diseases by targeting these RNAs?
That's the next frontier. The two main strategies mirror the biology we discussed for diseases caused by an overactive miRNA.
Like in miRNA that's wrongly silencing a tumor suppressor.
Exactly. The approach is to use anti-miRs, also called antagomirs. These are synthetic molecules, often modified nucleic acids, designed to be perfectly complementary to the problematic miRNA.
So they bind to it.
They bind to it very tightly and basically soak it up, neutralize it. They silence the silencer, allowing the target gene, like the tumor suppressor, to be expressed again.
Makes sense. And the opposite approach, for when a beneficial miRNA is missing.
Then you use miRNA mimics. If a certain tumor suppressor miRNA is lost or downregulated in a cancer, you can synthesize that miRNA.
A synthetic copy.
A synthetic, often stabilized copy and delivered to the cancer cells. The goal is for this mimic to get loaded into the cell's RISC machinery and restore that lost silencing function, hopefully shutting down oncogenes.
It sounds so elegant in principle, but getting these RNA molecules delivered to the right place in the body must be incredibly hard.
That is the major bottleneck right now. It's all about delivery. RNA molecules are relatively fragile. They're negatively charged, which makes crossing cell membranes difficult, and they can be quickly degraded by enzymes in the blood.
And you absolutely need them to get to the right cells, right? Delivering a potent silencer to healthy cells could be disastrous.
Precisely. Off-target effects are a huge concern, given the potential for one miRNA to affect hundreds of genes. So a massive amount of research is focused on packaging these RNA drugs.
Like the lipid nanoparticles used for mRNA vaccines.
Exactly like those. Lipid nanoparticles, LNPs, are a leading approach. Also engineered viral vectors, polymers, conjugates, anything that can protect the RNA payload, help it evade the immune system, and ideally target it specifically to diseased tissues like tumors or the liver.
So cracking the delivery code is key to unlocking RNA therapeutics.
It really is. Safe, efficient, targeted delivery is where the field is pushing hardest right now.
Okay, so let's synthesize this. We started in the dark matter, the 98.5% of the genome we used to ignore, and we found it's not dark at all. It's where the architectural plans are written, carried out by these non-coding RNAs. We've seen the tiny, precise miRNAs acting like post-transcriptional snipers.
Silencing specific mRNAs in the cytoplasm.
And the huge, complex lncRNAs acting as nuclear architects, scaffolding regulators, and directing chromatin structure.
Controlling the very accessibility of the genes themselves.
What's the really big takeaway here compared to how we used to think about genes and drugs?
For me, a critical concept our sources emphasize is reversibility. Think about the epigenome.
The layer of control on top of the fixed DNA sequence.
Exactly. Those chromatin modifications, the patterns of miRNA expression, these are part of the epigenome. And unlike a permanent mutation in the DNA sequence itself, which is very hard to fix.
Epigenetic changes are potentially reversible because they're based on chemistry, adding or removing tags, guiding RNAs.
That's the key insight. They're chemically reversible, at least in principle. If we can figure out how to intervene, maybe inhibit an lncRNA that's wrongly silencing a gene, or deliver a missing miRNA, we might be able to reprogram a diseased cell back towards a healthy state.
Without needing to edit the underlying DNA code, you're fixing the software, not the hardware.
That's a fantastic analogy.
Yeah.
We're correcting the regulatory software. This offers tremendous hope especially for complex chronic diseases where the underlying genetics might be complicated, but the epigenetic dysregulation is a key driver.
It shifts the focus from managing symptoms to potentially reversing the disease process at a fundamental level.
That's the ultimate goal, and
Ah.
connecting back to that idea of leverage.
Right. One miRNA hitting hundreds of targets.
Consider the implications. Traditional drugs usually target one protein, one enzyme. It's a single point of intervention. But if you modulate one miRNA or one lncRNA that controls an entire network or program of hundreds of genes,
The therapeutic impact could be vastly broader and more fundamental for complex diseases like cancer or fibrosis or neurodegeneration.
exactly, you gain leverage over the entire system by adjusting a master regulator. It suggests a future medicine that's less about fighting individual downstream consequences and more about correcting the cell's core programming language, the regulatory instructions.
Accessing the control panel, like you said earlier.
Accessing the control panel is a paradigm shift. So as you think about this incredible layer of control, this hidden world of regulation, what aspect of it, this vast network of non-coding RNAs, really stands out to you as maybe the most exciting avenue for the future? Where do you see the biggest breakthroughs coming?